Good afternoon, everyone. I'd like to thank ARM for the opportunity to speak this afternoon, and also to introduce, I was the former president. As of last week, I have changed roles now to be the clinical therapies advisor for the company. Formerly, I was the sort of chief clinical officer of Cesca. We are a publicly traded company. We do have forward-looking statements here, so what I'll be talking about today fall within the safe harbor area for a publicly traded company. Very quickly, for a corporate snapshot of the organization, we have about $16 million in annual revenue, around 100 employees that are stationed in two centers in Northern California, one in the Bay Area and one in the Sacramento area, and then about 50% or so of our employees in New Delhi that serve as our CRO and back-end clinical trial group. We did recently complete a private placement of $15 million. That was focused in two tranches. The first tranche has come into the company in $5.5 million, and it is they are debt facilities. We will have a second tranche closing on the notification from the California Institute of Regenerative Medicine grant notice for our pivotal CLI trial, which I'll talk about a little bit later. I'm going to start with the telescopic view of where we see our platform technology going in the future. We see multiple specific indications that we can target autologous cell therapies for, whether they be done in a banking platform or in a point of care or bedside, or even in other GMP facilities. Our process is amenable to all three of those. And, obviously we don't have the bandwidth nor the cash to do all and address all the $16 billion of opportunity, so we have started to focus. In 8 indications, we have human clinical data. We are now focusing on 3 of them. Our nearest commercial opportunity is in critical limb ischemia. We have completed 2 phase 2 studies in CLI, one with an intra-arterial delivery of a bedside produced autologous bone marrow derived product, and the second one a intramuscular delivery of a bone marrow derived bedside produced cell product. The intramuscular data was by far the most compelling of the study, and I'll go through that data in a moment, and that's the one we are pursuing and have FDA approval now to move into the phase 3 trial. Our other indication, and I'll give some discussion on today, is our Surgeworks platform, which is the newest upgrade to our cord blood and bone marrow processing systems. And we're also repurposing those and repositioning them for being available to bone marrow transplant across all of the GMP hospitals across the globe. We're different than most of the organizations that have spoken at the conference so far. One is that in the U.S., the FDA has determined that we are a device and not a drug. That means that our commercialization strategy and really the development strategy is a 2-phase clinical trial approach. So we have a phase 1/2 that's required where we look at safety and preliminary efficacy, and then that is always designed to get us into a phase 3 approval where we look at large-scale, statistically significant safety and efficacy. So we're pretty pleased with that. That shortens our development time for an individual therapeutic target by about 3 years. And so it just gets us closer to commercialization than had we been put into an IND pathway. Our product is called the Surgeworks and VXP system. It is a platform-based process that is modified for each of the 8 different indications I showed on the earlier slide. The modification includes all the various devices that are used to process and to deliver the cells into the different target organs. And as well, we have onboard diagnostics and cell processing that's all done right in the operating suite. The system is a platform that rolls into the operating room on about the size of a gurney. Everything the physician needs to prepare to capture the cells from the bone marrow, process them, test them for both purity and potency and redeliver them into the target organ in the 90-minute platform is all provided on a mobile equipment platform that rolls around. One area we started in this almost a decade ago was realizing that autologous-based cell therapies were being applied in the hospital setting probably inappropriately, meaning that cells are very fragile, they should be handled in a delicate way, and physicians were aspirating these cells under very high delta Ps and then re-injecting them into patients through standard off-the-shelf syringes, et cetera. What you end up at the end of that result is a very low potent, essentially dead cell product or on its way to being dead in a couple of hours. We've re-engineered the whole system and every step from aspiration through processing, including the diagnostics and delivery, are all specialized to the therapeutic and engineered around them. So what you end up getting is a disposable kit for each indication that is universally applicable on this one set of capital equipment. The cell processing that we use is a very sophisticated process that the surgeons can do. They aspirate the bone marrow, put it into a disposable bag set that then locks into the device in the upper right-hand corner. That device is put under very high-speed centrifugation in the operating room on a mobile centrifuge. And at 1/1000th of a second, as different cells go by, they are shuttled into either a therapeutic bag or shuttled into the waste bag. And then at the end of that, the physician takes off the therapeutic bag and prepares to deliver them into the target organ. The mechanism of action primarily for our focus is in revascularization for ischemic diseases. And I think the target is a multi-cell approach is better, in our opinion, than a single-cell approach. The reason for that is that we ultimately end up needing to have multiple growth factors and multiple cells present to form vessels that reperfuse the target ischemic area. Focusing on our near-term critical limb ischemia, we are focused in the high unmet need of no-option patients. This is a huge epidemic across the globe. It is becoming an epidemic in the U.S. as well. But when we look at the no-option patients, and even for the advancements in things like endovascular atherectomies, stenting, and balloon angioplasty, still over 70,000 patients per year have a major amputation due to critical limb ischemia. And that is our target population. So it's still large, even with all of the advances in care. If you add on the minor amputations that the patient will have leading up to the major amputation, it's another 134,000 procedures per year. So you end up with almost 225,000 amputations on CLI patients per year, and a big economic burden to the Medicare system and the overall economy. We started out looking at this in animal models, then translated it into in vitro models, and then took it into the human data. Suffice it to say, I'll start at the very high level. All of these patients had no options. They were recommended for amputation. We put them into the trial, 17 patients, and at the end, 82% of them had limb salvage at 12 months. Most of them would have had an amputation within a matter of weeks, if not days. So it was a very unexpected result and a much higher result than had been seen in this indication to date. Off of this data, the FDA approved us now to go into the phase 3, which we are preparing to get underway by either the end of this calendar year or very early the next calendar year. It'll be 224 patients, and it's an adaptive trial design, meaning we do an interim analysis so we can actually repower the study in the middle if we start to see the placebo arm have higher than expected responses. Our second near-term indication is AMI, focused on ST-elevated AMI patients who do not respond to reperfusion therapy. Reperfusion therapy is either stenting or a clot-busting drug. And, surprisingly, about 10% of the patients do not respond to these. And the response is pretty easily predicted in about 72 hours post-reperfusion, whether that patient is going to progress pretty quickly to chronic heart failure and then ultimately have a very high mortality rate. The mortality rate curves are shown there for non-responders, and essentially at the end of 4 years, half of these patients that have not responded to reperfusion therapy are dead. And that is our target population for our therapy. We intervene early on in 3 to 10 days after the MI and after reperfusion has failed. And our goal is to reduce the adverse remodeling that happens post-MI in every patient, and ultimately to measure the change in ejection fraction in that patient after 6 and 12 months. And if you measure them and you look at the long-term studies, which has been shown in a large global study called the Cadillac risk score, ejection fraction is the best predictor of both long-term and short-term comorbidities and mortalities. And in the U.S., nothing drives us more than looking at the economics of chronic heart failure, which is rehospitalization while the patient is still alive. And so we're pretty excited about this therapy, something we've been working on a long time, and we are in the process of preparing the application for the phase 2. The phase 1 was a single patient that came into the ER, had a dramatic ST-elevated MI and an ejection fraction of 36%, did not respond to stenting. We treated him, followed him for 24 months, and his ejection fraction came back to 60%. If you look at the large studies that have been done in Europe, and there have been almost 1,000 patients treated with intracoronary bone marrow-derived cells, you see in this low LVEF responding patients that they do very well on stem cells. So if you select your patient appropriately, we're pretty bullish that the cardiac studies are going to start to show very promising results. But you need to be very selective of who gets treated. Our study that we are preparing to start now will be a 40-subject randomized active control trial, 1-to-1 randomization, and with the primary endpoint being safety for MACE and the secondary endpoints being volumetrics and assessments. The third indication that we're focused on is the bone marrow transplant program. Recently, we've announced 2 collaborations in the U.S. One of them last week was St. Jude, the second one being with Memorial Sloan Kettering in New York. Our focus here is to give our rapid cell purification systems, make them available to hospitals to do very tricky bone marrow transplant procedures where they need to have high stem cell recovery but eliminate some unnecessary cells. 2 of our programs are one focused on adverse red blood cell mismatch, and the other one is on a white blood cell mismatch where we actually remove the T cells from the transplant unit itself and then transplant the stem cells. We are in clinical trials with both of those and collaborating with those 2 companies I mentioned. Finally, how we get our clinical data quickly, we do all of our phase 1 and 2s based out of India with our partner, Fortis Healthcare. It saves us a lot of money, and with Fortis, we're able, the hospital we're embedded in was just ranked number 2 in the world for being the most advanced hospitals in technology, and this has also been vetted now by the FDA, giving us approval to use these results in an advanced clinical trial. We have a lot of near-term milestones for the company this quarter and next. I won't go through each one of them, but they are listed here. And we have been having a reasonable number of press releases recently, and we anticipate having more of those. So kind of wrapping up, we have treated over 20,000 patients with our platforms, so we understand the safety profile of it. 2,500 pediatric patients have been treated in our cord blood-based program. And so it's just a well-established, stable platform overall. We have a good IP portfolio and a good, robust therapeutic target. And the final note I will make is that we are working with CMS on our reimbursement rate, and by being designated an IDE, CMS will actually reimburse our patient costs for phase 3 trials. So we're in the process of submitting that application for CLI, and that will reduce our need for dilutive funding by between $4 and $6 million for that trial alone. So I'll thank you for the opportunity, and if anyone has questions, I'll be happy to meet you outside. Thank you.